I'm going to step way back in this talk. Every single person in this room, regardless of what field you're in, has wondered, are we alone? That's sort of why you have this institute. So we've all wondered that. And I think in particular with the ever-increasing number of exoplanets, this question, and Earth-like exoplanets becomes even more prescient. But as we all know, it's an N of 1. And, you know, Earth was once a rock hurtling through space without any life on it. And now it's still hurtling through space, but now it's covered with green scum, right? This whole thing we call life on the outside of this planet. But the great thing about being on planet Earth and having this particular N of 1 is that, of course, we can go back in time. We can look at Earth at alternative times. So we can go back in time, and we know that 3.5 billion years ago, it was a watery planet. We know that we had life at 3.5 billion years ago. This is from Western Australia, the just beginnings of wisp of life. And I'm going to fast forward about 3 billion years now. So we have this fossil record that goes back, and we have geochemical markers of life over the course of the history of the Earth, and we can go back and look at Earth at these different times. And that, in fact, informs a lot of how we think in terms of astrobiology. We also have the unfolding of animals on planet Earth, and that's what I'm going to talk about today. And it's very familiar to a lot of people in this room and perhaps less familiar to others. So I'm going to stand way back. But I'm going to talk about the sort of early history of life on planet Earth. And specifically, I'm going to talk about the Ediacara Biota. That's this fossil here, not that guy there. And just for the quick summary of what the Ediacara Biota is, some of you in this room work on the Ediacara Biota, so you can have a mini snooze. It's a suite of fossilized, soft-bodied organisms. So nobody has a skeleton yet. Everybody, when I say I'm a paleontologist, they're like, oh, dinosaurs. And I'm like, no, much earlier. And they just can't conceive of things that don't have a skeleton. So no skeleton. They are globally distributed, but a handful of localities. It spans about 30 million years. That is a long time in geologic time. Humans have barely touched, well, not even close, right? Order of magnitude less. I'm going to talk about the Ediacara Biota of South Australia today. There's actually a domestic stamp series that was made on the Ediacara Biota in Australia, so I have to always show it because, like, that would never happen in the US. Well, definitely not now. And so when I'm focusing in on the Ediacara Biota, it's where I've worked, but also it has a lot of firsts in terms of animal evolution, which of course impact this planet. And just, you know, for those way outside the field, the time period is called the Ediacaran. After sort of Ediacara Hills, where we work, it's called the Ediacara Biota, and I work on the Ediacara Member. So it's way too many Ediacaras, but just to clarify that. The other thing, just to give you an idea about these guys, they are small and big. People sometimes are thinking, oh, it must be hard, like first multicellular life. You know, are you looking for little things? They can be this big. They can also be millimeter in size, so pretty wide span. Soft-bodied, but still, given that they have no biomineralized skeleton, they are still very morphologically diverse, as hopefully you'll see. The other thing is they're really common when they occur. Typically very common. In the best sites in the world, they're common. You're not spending a day trying to find them and then like, woohoo, you know, they're everywhere. Here's the crazy thing. This shows you how weird they are. There's two things about them that show you that. One, most genera are monospecific. So a genus and one species, if you think about, like, I don't know, usually the example of oaks in the US, there's, you know, 50 species of a single genus of oak. So one genus species, that's very different on body plans. Also, these guys are so weird that we talk about genus diversity, but they're so different, and the body plans are so different that we don't even group them into families, much less orders, class, phyla, and so on, right? So it's this weird experiment, evolutionary experiment that happened on Earth. And the question is, how can we fit them into animal evolution? So this gives you an idea, not the bearded dragon down there, but an idea of some of these things. And some might look familiar, and you might say, oh, that's bilaterally symmetrical. But then, the middle guy up there has triradial symmetry. Nothing alive today has triradial symmetry, no animals today. So a lot of crazy body plans and experimentation. So as I mentioned, the Ediacara Biota spanned 30 million years. So this is time on the left and different types of organisms going across the bottom. And many in this room work on the Avalon assemblage, which is the oldest. I'm going to talk about this middle assemblage here, that's sort of the medium gray, where you can see every one of these lines represents a sort of group of organisms that there's an increase in diversity. Right? There's more types of organisms in that middle gray. And we call that the second wave of the Ediacara Biota. You have this diversification of morphologies. And that what we get in this time is the advent of sort of novel both behavior and morphology at this time. So my work is focused in South Australia at what is now Nilpena Ediacara National Park, and we acknowledge that this land lies within the Adnyamathanha traditional lands, and we respect elders past, present, and future. So we're going to step back 550 million years ago. You're snorkeling around. What are you going to see? So number one, we're in the marine environment. There isn't really life on land. In the marine environment, it's sort of nice, I will say like the Gulf of California, some other sort of pretty protected area. And there's things living on the seafloor, and every now and then there's a storm. And that storm comes and buries that seafloor. So here's your organism sitting on the seafloor. A storm comes in and dumps a pile of sand on that organism. And what happens is that organism eventually decays, but before it decays, this sand casts the top of the organism. So we have a cast of the organism on the base of that sand bed. So that if we look here, each one of these beds is a storm bed, and the fossils are on the base. The casts of that living environment are on the base of that bed. So to get at those fossils, we can excavate these beds. Now, most paleontologists, really none in this room, but most paleontologists do what we call the clink-clink method. Clink, 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 take it back to a museum, right, and study it in the museum. What we do and what paleontologists here, many do, is actually have the advantage that you take that ecosystem, you dump a pile of sand on it, they're all preserved in situ. So if you can excavate that bed of sand, flip it over, piece it back together, you're looking at a cast of that whole seafloor and everybody where they were living. And that's what we're aiming to do. When we first started this, we were going to take stuff back. And we're like, and this is with my colleague Jim Gailing, I wonder if we can excavate these beds. This is not something that was done before. These weren't exposed. So we're like, let's try it. And the person who owned the cattle station was like, yeah, try it. So we did. It is totally laborious. So much of my time and my students' time has been spent doing this. And there's a whole way of doing it. And the amazing thing is no rocks or fossils were harmed in the making of these reconstructed beds. The beds are sort of dipping gently on a slope. By the process of uplift and turning them a little bit way, these beds all are naturally broken. And some of the pieces are the size of your dining room table, and those are really heavy and hard to move. And some are the size of your pinky nail, and those have their own complications. So we didn't break these rocks as we put them back together. So I came here with my colleague, Jim Gailing, and this was my kids' the second year. And we had an NSF grant to do 2 years in South Australia and then go to Namibia. So this is my kids today. I'm still working in South Australia. And I say that to the students in this room who think, oh, this is my path and I have this, set and I'm going to do this and this and this. It never goes that way. If you had said to me when my kids were this age, you're going to spend the rest of your career in South Australia and it still won't be enough, I'd be like, oh, no, 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 no. So got that wrong. Still going back. So what we've done is we've excavated these beds. This bed is that you can see people here for scale. These are post-its, about this big. We've excavated about 40 beds. The size and shape of the beds is dictated by the geology and a few other factors. I started off, and I was telling the story to the graduate students, my first question was, would I have needed fins in the Ediacaran? Thus the 2 years there. And I thought, we're going to excavate these beds. It's all going to be much of a muchness. And so fins is like, you go to the Great Barrier Reef or someplace else and you need fins because you're like, oh, look at that. That's different. And I was like, totally not going to need fins. So got that wrong. 40 beds later, we're still turning up completely new body plans and whole different combinations of things. So again, think this is, early on, Earth's first ecosystem, right? The whole span of the Ediacara Biota. And we already have lots of different communities and lots of complexity. So we excavate these beds. We map them, we LiDAR them, we do scans, we do photogrammetry, we do all sorts of things, but we also get on our hands and knees and post-it everything we see, and these post-its are color-coded to the organisms so that while we are there, we can actually get a sense of what's there. So what do we find? And again, with the idea of first, like the fins thing, okay, we totally needed to do that, but these weird organisms, and how are we going to get at them, right? They don't have characters that are easy to come by. So I will tell you the story of the very first thing I described from this. So this is a long time ago. It was described as Form A. It was initially described by others as fecal pellets. There should be so many reasons that everybody, even the non-biologists, are looking at this and say, no, right? Branching, I'm not going to go into it, but think about it. Lines of these guys, right? So many reasons. The thing of it is that when you have whole bedding planes, you have tons of specimens you can look at and compare. And it allowed us to see a lot of this particular organism, which we called Phoenicia dorthea. And we interpreted it as the body fossil of a tubular organism, so something that stood erect on the sea floor, and that what we're finding is them collapse down. And these are just a lot of different pictures, mostly of that. And then we found their holdfasts, where they're attached to the seafloor. And what we realized when looking at the holdfasts at the time was that they were densely packed, that this particular slab I love because it has two different sides right next to it, that they had cohorts. So in that dense packing, they're all the same size. And here you can see little ones, here you can see bigger ones. We would see this with the actual Phoenicia themselves, but easier to see here. And so this is a case of what we would call synchronous aggregate growth. Synchronous, single cohorts. Aggregate, they're all stuck together. And growth, this is how they grow. So that kind of growth is what we see with sexual reproduction, a product of sexual reproduction. It is a case where, and this was my next slide, you have egg and sperm triggered by the environment, the moon, whatever, seasons, put into the water column, and you have this cohort. It's very effective. It's not what we would have expected in Earth's earliest ecosystem, because you do it for things like making sure you have a hold on the seafloor or gamete wastage and all those other things we interpret from modern organisms who do this. But here it is in its early ecosystem. I was telling this story earlier. Today, we published it in Science. Basically, second half of a sentence was, this is consistent with sexual reproduction. That took off. If you can read this, as I was also saying, it was in the Daily Mail, page 3. And named after my mother, which was even funnier. So a lesson in press, but it was fine. But just how do we get at this organism? Not because we can see all these characters, not because we can look at it and say, oh yes, but going after, in this sense, coming in from the angle of reproduction, which for some of these we can do, other ones we can't. But in this case, we could sort of get a handle and say, all right, it's Cnidarian grade. We're not going to say that it's Cnidarian, but it's consistent with that. Oops, sorry. So sorry, this is it's my own computer and it's sensitive. So just stepping back, we're looking at these surfaces, right? And as we look around, the surfaces look like this. Okay, so if you are in physics or astrophysics or worry about things like that, you're like, yeah, I don't know what she's talking about here. They all look a little bit mottled, right? You're sort of, that looks like, that's called weave. There's this actually looks kind of like wet sand, right? What all of these represent is the top of the seafloor. And you can see they're sort of varied, but they all reflect organic mats that covered the seafloor at the time. And nowadays, we don't get them very often. And that's because we have things like this. This is a sea cucumber. It is the size of a family loaf of bread. It's huge. And why I show this is because these guys are really effective at eating absolutely every bit of organic that is ever on the seafloor, as do everything else that's eating all the organics on the seafloor, right? It's like a buffet. And so the thing about the Ediacaran is we also didn't have organisms like this. It was just the first whiff of mobility, as you'll see in other slides. So instead of a seafloor looking nice and clean sand, like we have it today, they would have looked something like this, right? It would have been disgusting. You would not have wanted to put your foot into it. And it would have been variable and made up of consortiums, not just of prokaryotes, but prokaryotes, eukaryotes, certainly multicellular things, covering your seafloor. So as you think about your, diving in the Ediacaran or snorkeling, you've got your fins on. There's lots of weird things on the seafloor. The other thing to know is, okay, there's a microbial mat or an organic mat covering the seafloor. That turns out to be really important for a lot of reasons. Integral to the ecology. Everything living in this organic mat is interacting with this organic mat. And that's a really important piece of information for us. And so we can look at that interaction and get an idea about life modes and behavior. And I will end with talking about the impact of those mats on sedimentary packaging and how we even think about what happens in environments when you have organic mats. But what we have during this time period is we have the advent of motility, so active mobility, things actually moving on the seafloor. And that is a major breakthrough in the evolution of animals. We all love to go out to dinner and be served. But if that was the only way that we could get food, right, we'd all starve. The ability to move, to go search for food elsewhere, to go to a better environment, a nicer organic mat, huge evolutionary novelty. And what we see during that middle gray period, during that Ediacara Member time period, is the advent of mobility, active mobility. And the way that we see it is because organisms are interacting with the mat. So your seafloor is covered with this green scum. And it's the way that organisms interact with that green scum and eat that green scum that leaves a record for us to see. So that's a burrow, right? That looks familiar. You could see that in any modern tide pool or whatever. And it's something called helminthoid ecdysozoan. And we've known about it for decades. So we've known there was somebody that squidged around in the sand, making a burrow like that. We know that whoever did it could go, remember, seafloor is covered with microbial mat. It's eating the microbial mat. It's interrupting the sand as it goes through it. It also will track that sand under very thin, track the organic mat under very thin sands. And we can see that because we see the burrow on the bottom of sands. That's a really advanced thing. We also can see from the burrow that it did peristalsis, like you see worms move today. So we've known about this burrow for decades. We're like, certainly early bilaterian, generally accepted that whoever made this was a bilaterian. Our closest relative, we're bilaterians, bilaterally symmetrical, through gut, head, anus, and so forth, right? So we know this thing exists. We have its burrow. We know it ate mats. I don't know why the lobster came on separately, but also lobsters. Always wondering, were we ever going to find who made it? But it's going to be really small. Those burrows are a couple millimeters across. And then we excavated a bed and found this, right? Which looks like a mini rice grain. And if you look at that and say, we did too. And then we're like, actually, we should look at this a little bit more closely. So we got a scanner and two very happy looking people out there scanning. Got a laser scanner so that we could actually look at these little guys, right? This is about 1 millimeter by 2 millimeters, so really small. And remember, it's the cast of something, right? So we did that and found this, which we named Ediacara, and it has anterior, posterior. You can see that's a little higher than that end. Associated with burrows, this bed turned out to have hundreds of these guys on it. It is modular, so you can see, you know, I don't know that it was purple, but that's my signature color. So you'll see every reconstruction, they're all in purple. It's just because my students know that about me, so they always make it purple. So it's modular. Presumably, you can see some of the modules here. And so we have this early bilaterian. It occurs in the absolute oldest part of the Ediacara Member. So it's really down there in terms of even our period of time that we record. It's not the only thing that moved. This is another guy that moved. This is Dickinsonia. This is my former student, Scott Evans, who did a huge amount of work on Dickinsonia, looking at Dickinsonia growth, anterior, posterior end, and so on. But one of the things about Dickinsonia that's interesting, and that's about this big, that specimen, is that it moved. And we know it moved because it left what we call footprints, and it's the worst word ever, but it's been called footprints for ages, so we just lean in. So if you look, there's a Dickinsonia. It's sitting on the mat. What Dickinsonia did was it absorbed mat through its underside, right? So very few things do it today. Placozoans do. And we don't know exactly how it did it, but it absorbed nutrients through its underside. It would then move, and where it had eaten, it would leave a divot in the mat, right? And then a storm comes, and it fills in that divot and casts the Dickinsonia. So the end result is, on the base of the bed, remember, a positive and a negative. Okay. Footprints, they're not super common, but we find them. We have a trackway of 13 footprints. We can tell with the Dickinsonia at the end, we can tell the direction it was moving, and so on. So we know this guy moved, and it was pretty clever because it moved, and it would go maybe a centimeter, like if they're this big, which sometimes they are, it would go about a centimeter past the last one, centimeter past the last one. So this wasn't like hibbity-jibbity movement. This was, you know, actually having some sensory enough to know, oop, I ate that, I haven't eaten this. But I'm not going to waste so much energy going all the way over there. Tells us a lot about this organism. So we know it moved. So we can look at this. Again, this bit around here, that's made by organic mat, the sort of rough texture. But what you see when you look at this is it has a really sharp edge around it, right? But think about it. This is some guy sitting, or gal, sitting on the mat, right? It moves pretty often. So when a storm comes, its body on this mat, that's going to be a pretty sharp boundary, right? It's just newly there. It hasn't been sitting there for a long time. And that's opposed to things like this, where this is Obamas, which we named a couple of years ago, sort of a torus shape. That's a centimeter for scale. And when you look at that, kind of hard to tell the end of the organism and the beginning of the mat, right? But you think about it, this organism, then we interpret this as being sessile. It didn't move. It's sitting in the mat. It's not moving. So mat's going to grow up around the bottom of it, right? Same with this guy. This is pretty small, so it doesn't have such mat. But again, this is a silly putty, because remember, this is a cast, so we use silly putty all the time to see what the organism looked like. Same with that. Not that sharp boundary, right? It's so subtle. It's so like, are we really talking about boundaries of fossils? But that is a way in which we can tell whether something was sitting in the mat for some period of time or moving. So this guy here is now called Amberites. I found it on 2 beds. You know, the first 35 beds, I had 2 of them. I logged it in my notebook as raisin, because it kind of looks like a raisin. I thought, it's going to be some smush Dickinsonia, but it was different. When you look at it, really sharp boundaries, right? Like other things that are moving, but still, raisin. Didn't want to deal with it because I'm like, is it something? Probably not. Till we had a bed that had about 100 of them, and then you're like, okay, now I have to deal with raisin. This is my student Heather McCandless, who's been working on Attenboroides. So this thing, very sharp. So you know it's not just sitting there on the seafloor. It's not been there. It's not overgrown with mat, right? So that's observation number one. Observation number two, there are a lot of different shapes. They're all, sort of pinky size to pinky nail to thumbnail size. This, again, centimeter for scale. Largely sort of oblong, but never really regular, sometimes circular. And then they have what appear to be ridges. There is no rhyme or reason to the ridges. They generally go parallel to the long axis, but there's no correlation between size and number of ridges. There's all sorts of things and all sorts of measurements we've made. The conclusion of all of that is that these ridges are folded tissue. And so Heather took all of these, she scanned them, she then calculated the surface area, and we're only seeing 1/2, we think. I mean, obviously, 1/2. There's no, also, this is the closest we get. Is that a sharp edge? And it actually is. So there's not, it is sharp all the way around. So she did that and then used the 3D software, used Blender, which you all would know, and was able to reconstruct these and basically blow them up because she knew the outline. We had constraints, right? And we knew the outline. And so able to blow them up and looking something like this. Looks kind of like a ctenophore. I'm not going to say it's a ctenophore, but it looks kind of like a ctenophore. And what we see is that we put it in the water column. Why not is it something globular rolling on the seafloor? All right, there's things that are globular that roll on the seafloor. One of which is its spatial distribution is random. You might expect it to all be clumped in one corner. The fact that it only occurs really on one bed and with huge amounts of seafloor exposed of the same facies that we only get one occurring someplace else. There's no it doesn't, like, it doesn't it doesn't explode out one end. I'm sure there's absolutely a better way of saying that. Because that sharp edge goes all the way around. So we think that it deflated. Lots of times these little algae will roll around on the seafloor, but when they compact, they actually sort of explode out one end. These guys do not. So we think it deflated. And, not to take the raisin analogy too far, but to take it probably a little bit far, that it is a grape and that it actually, but it wasn't, but that it deflated upon landing on the seafloor and compaction, and that when you blow it back up, it looks something like this, and we interpret it to have been pelagic and in the water column. That's the most parsimonious with the data that we have. And a lot of things in the Ediacaran have been interpreted to be pelagic. One by one, they all end up being benthic. But this one so far has stayed pelagic, and we feel very good about this. So we're not surprised that there are things in the pelagic realm. There should be. In terms of macroscopic animals, but this is then one that we would put there. So going back to the sharp edges, this guy here, it looks very blurry right now, is Sprigina, very sharp edge. Everybody looks at this and they're like, I'm going to find a Sprigina trace fossil. Surely it looks like it has a head and it's bilaterally symmetrical. But some people have interpreted this as a holdfast and flip it this way, Dolph Seilacher. What we would say is that, first of all, very sharp boundary. It's not hanging around on the seafloor. Also, if you're moving around, unless you actually impact that mat, unless you actually are like really digging into the mat, your trace fossil isn't going to be preserved. Your burrow isn't going to be preserved unless you actively disrupt that mat. And so we would say we are very confident this guy moved. Also, just the way it manipulates its body, it often is preserved with a little bit of a crook, which would be consistent with being in the water column, or just even not in the water column, really just sort of right on top of that sediment-water interface. So it's, again, using that sharp boundary, which, as I said, is a little bit of a theme in this talk. So we've recently described a couple of taxa. This is a new thing called Quaestio. And when we found the first one, we thought, oh my God, we're seeing things. Because if you look at it, there's a question mark, right? And you're like, come on, it's late in the day. You're like, this must be something weird. It has a little posterior pinch. They have footprints. This isn't the best footprint, but the positive, but it's right next to it, so we keep it. So the first one we found, all right, this is something weird. We're getting some taphomorph. There's something like that. And then we found on this bed here, which is called Fun Bed because it's so much fun because we find so many things. And this is the group that are known as the Nilpena Pit Crew, who have been excavating this bed. So this Quaestio, this is its. I wasn't kidding when I told you about the purple. This is the reconstruction. Why it's important? Totally weird body plan, right? And we have 11 specimens. They all have this question mark. Why is that interesting? It is, so Quaestio for question, Simpsonorum is after a colleague. It's still in that category of early motile organisms, things that are moving. It left footprints, but it had something really interesting that we haven't seen in any of the other motile organisms. And that is this asymmetry, this what we would call chirality. We are bilaterally symmetrical, but actually we're not 100% bilaterally symmetrical, right? Our heart is on one side and so on. So this sort of weird left-right distinction, which is actually really common even in things we consider bilaterians, puts this character way back in the Ediacaran. Now, what this was, I have no idea. So if that's going to be your question, the answer is I don't know, so I'll put that out there right away. Just for that whole media thing we were talking about with this was, if you have an iPhone, was one of Apple News's conversation starters. When, it's like purple blob, what do you think? And it's like, anyway, we didn't put it there. So this is one of the things, this is Fun Bed again. So we're, in the South Australian outback. This is a fossil that we found. This was the first specimen. The nickname was fishhook. Again, we weren't sure it was going to be anything. And then we found, about 150 on this bed. So early on discovered it and was like, then found a lot and thought, once again, I have to deal with this. Things to note, and here's what these guys, so this scale, this is a 1 centimeter scale. This is a bunch of specimens, and these are the laser scans. So things to note, very sharp, distinct boundary with the mat. No evidence of modularity. So remember, Acaria had modules. This is smooth completely. When we look at all of these specimens, they have a sort of, can be sinuous. They can be a boomerang. They can be hooked. They never have tight folds or hooks. They never come back on each other. They do have anterior, posterior differentiation. One end is always a little bit bigger than the other end. This is just, showing that. And what's crazy about these guys, and again, remember, we're trying to figure out, this is a line in a rock, right? And people are always like, this is a line in a rock, Mary. Incredible relief, right? So this is the cast of something that was on the seafloor. And you imagine, you've seen Attenboroughes, we have lots of tubular organisms and things like that. They collapse, right? Imagine an earthworm, right? It's going to collapse pretty quickly, right? This guy, in terms of preserved relief, is one of our highest. Basically, we are preserving a circular diameter. It compresses a little bit, but per sort of length and diameter that is, sort of length and width, we have an incredibly well-preserved diameter. This guy, when it was, when the sand came down and smothered it, kept its relief really well, almost perfectly. So for us, we look at this and talking to lots of ecdysozoan people, which is, for those of you who, that isn't the word you know, that is the clade that includes all of insects, crabs, arthropods, and things like nematodes. This high relief, we think, is consistent with it having had a cuticle. Associated with it are these trace fossils, which are quite sinuous. So trace, this is burrows, again, a centimeter. So this is the record of it moving through the top, moving through the microbial mat. And so in the end, we have interpreted this, these sort of worm-like fossils to be the oldest known member of ecdysozoa and possibly a nematode. And I was telling the group today at lunch that we have a department of nematology, which is the only one in the US at my university, and they love this. They're like, it's a nematode. We love it, love it, love it. It's bigger than most nematodes. Most nematodes are really, really tiny, but there are nematodes that are this big. And they're very happy with this being a nematode, particularly in the manner in which it is preserved, that sort of, nature of sinuosity. This is not our reconstruction. This is one of these weird Wikipedia. So somebody read this paper, sent me an email, Kevin the cat, sent me an email and said, I read your paper, I thought you needed a reconstruction, so I hired this guy to do it for you here. Right? And then I'm like, deep dive on who is Kevin the cat. Have never found out. I wrote him and said, thank you, thank you. Right? It's a really good we're very happy with this. So this then got put on the Wikipedia page. We had nothing to do with it, the Wikipedia page or anything like that. So one of those really kind of magical, but a little bit creepy things that came out of this. But, you can't use it without telling the story also. It's not purple. So you'd know that it wasn't one of my students. Okay, so that is looking at individuals and how those individuals are on the mat and so on. But we can also look, we have preserved whole communities. And so we've done a lot on that. So in terms of what can we say, and these are just a few examples of how we sort of get at unraveling, sort of how do we untangle this early ecosystem where we really have no idea what these things are. You guys hear it from Emily and others in this department, that the early evolution of animals is in the Ediacaran. I always say not popular with Cambrian workers, but by the time you get to the end of the Ediacaran, everything is just fiddling on a theme. But that is not that popular, but it's popular with some. That our ecosystem is far more complex, and I haven't gotten into a lot of that, but just even things like commensalism, far more complex than we might have imagined at this time, and that really when conditions were right, life took off. Right? This is a really big diversification of all sorts of different things. So I'm going to switch gears massively right now, because my whole thing was search for life. That was the end. Yes, we can search for life, and I know this group thinks about things like that. So we're going to think back to this whole mat world, right? So I am absolutely convinced we're not going to find Dickinsonia on Mars or any other planet or any of these other organisms. Like, I don't think I'm going to say that. Microbial mats, though, are an interesting thing, right? And we know, here's your seafloor. It's covered with microbial mats. It leaves a signature. You guys have seen some of that. And it doesn't matter that you can't see this. This is a list of just how you might look at a rock and where you might see evidence of microbial mats. Because remember, microbial mats, we've had them for, not remembered, we've had them for a long time on Earth, sort of 3 billion years. And they've gotten more complex and are way more complex in the Ediacaran, but they've been around for a long time. But they impact the sediment. You can't help that, right? You have a whole seafloor and it's covered with a microbial mat. That's going to impact the nature of that sediment. So one of the things we've looked at is ripples. Now, these are exposed ripples, but it's like such a nice tidal flat that I had to put it in. We're submarine, so we're underwater, right? So that's the first thing. We're underwater, but if we think about the seafloor today, even on that tidal flat, if you were standing there and the waves come in, it's going to erase all those ripples, right? The next storm that comes in, the next tide that comes in, right? The same thing happens if you're snorkeling or diving and you're looking at the seafloor. You see sand migrating across the seafloor as a storm comes in. in the Ediacaran, particularly in this little area of the Ediacaran that is pretty protected, we have a very specific, very weird kind of thing, which we call palimpsest ripples, right? So you have sediment deposited, you have a ripple top, and it gets covered with a mat. And what that mat does is it stabilizes those ripples. So when the next storm comes in, instead of reworking those ripples, as happens today, and since we've had animals burrowing in the sediment, that sand just casts on top of it. Even if it's the same grain size, even if it's a little bit coarser, instead of reworking those ripples, it casts on top of it. So you get these, sort of double ripples. And you can get them later on with clay, but in this case, it's literally sand on sand. And that wouldn't happen in the absence of this microbial barrier. So based on this, we were like, this is a sign of life. This is a biosignature, right? This is something that we could see on another planet. Were there microbial mats on another planet, and so on and so forth. So we got a NASA grant, and I teamed up with very clever AI people in our engineering department, because they've always like, can't we do something in our sciences? And we developed a couple of techniques. So it's all image classifier. So it's machine learning. How do we classify these photos? So the idea being you get a ton of photos downloaded from Mars or, another planet. And the first cut can go through without human eyes looking at it. So this is just a classification method. So this is Birbanu Padmaja, who's the very brilliant engineering graduate student who did all this with me, and Rachel Surprenant, who is a former graduate student. So the first is the classifier, and I didn't do any of the coding or anything. This is all these guys. But, with the idea that first thing you want to do is classify, is this an area of interest? So this is the Wanaka Formation, which some of you will know, so the older formation in South Australia. Heat maps, this is Mars. This is curiosity photos. So what our heat map shows is, hey, that's an area of interest. Right? So the first cut is developing this classifier that says, is there even an area of interest? So if you guys keep up on, sort of Curiosity missions and photos coming down from Curiosity and Perseverance, Curiosity has found a bunch of ripples. So that was really handy for us. I am not going to say there are microbial mats on Mars, so just making that clear. But this would say there's area of interest, right? And then on the right, these are areas that are not of interest. So, this is pretty good, right? It's sort of saying, yep, up here, not interest, down here, not interest. Of course, there aren't any rocks there, so, but we're getting there. We're still working these through, and that these are areas of interest. So you have a classifier that might say, hey, take this through your next filter. And then we have another computer vision program that is also sort of based on image analysis to say, can we, and I'm sorry that does look a little blurry, can we recognize these palimpsest ripples? Can we recognize them through a series of photographs? And so we have a couple papers on this that have been published. Very fun, crazy fun to work through this because things like cracks and shadows become so important. So this is a couple years in the making. We're now at the point where we will go and test this on other rocks of Ediacaran age and actually other places where we have the Ediacara Member to see, if it actually takes the next step forward. And as we think about sort of biosignatures and life on other planets, it is a story of thinking outside the box, right? I do a lot of NASA stuff, and nothing is about now. It's all about 30 years from now, right? What are we going to be thinking about 30 years from now? What mission are we planning? This is, do I think this is going to be on a rover anytime soon? No, or ever. It's proof of concept. It's, hey, we can start thinking about these. There are ways to use, Mars analog or even other planet analog on Earth to sort of address these and go through types of methods. So that is, again, we're not going to find an Uncas on Mars, and we as far as I know, we're not going to find palimpsest ripples as far as we know right now, but it's a way to start thinking about those kinds of things. So with that, just to advertise, this is a new national park. So we've been working there for 20 years. It is now a new national park that any of you guys can go visit and get a tour of with a great AV experience. And just because it's such a pleasure working with the Australian government. We are in for a it is National Heritage Site, going for World Heritage Serial Site. That is the GSSP for the Ediacaran Period, for those who think about that. But serial site throughout areas of sort of the Flinders Ranges, South Australia, for the dawn of animal life. And we are the 2026 Australian submission, which is terribly exciting. So with that, this is lots of students. Those work I showed you pictures of, but lots of students and mostly huge numbers of people to excavate beds and clean beds and all of that, and probably 20 years of funding from NASA. So I will leave you with this, which is the view of the night sky over one of our buildings at Nilpena Station. With that, thank you very much.